Welcome to our poster presentation. My name is Ioannis Giannoulakis and I am a postgraduate student on bioinformatics at the School of Medicine of the University of Crete. And during this period I am working on my master thesis in collaboration with the Institute of Computer Science of Forth, and the title of this project is a graph representation of the individual exome variation with evidence from biomedical text corpora. One of the most commonly performed genetic tests is exome sequencing, and it typically results in about 50,000 variants. If we could look up the information regarding these mutations throughout literature, it would be a huge advantage for the field of clinical genetics. This project deals with an approach on how the word of text entity can be combined with the exome sequencing word, and its objective is the extraction of a subgraph from the complete biomedical corpus containing information relevant to an individual's exome. In order to achieve this, we used PubDator Central Collection, which contains 30 million abstracts and 3 million full-text articles. PubDator is a service that is hosted and curated from NCBI. These articles are annotated concerning genes, mutations, diseases, and chemicals contained in the articles. These result from various open-source tools such as TMVAR for variants, GeneOR for genes, and Tiger1 for chemicals and diseases. And these articles are in XML format, which is a simple format with multiple annotations. So we parsed PubDator collection and organized entities into a graph database. We used Neo4j, a graph database that allows organization, data connection, and enables queries. Neo4j uses a declarative language for defining, inserting, and querying graphs. And our graph contains 22 million nodes and 111 million edges. And once a user downloads the biopsy corpus, the time required to build the graph, which contains more than 100 million edges, is less than 10 hours. Here is a video example of querying on the graph database. This is a query for a specific article. It returns a node of this PMC article, and if we expand this node, we can see all its contents. Genes with the red color, mutations with light brown, and a lot of chemicals with yellow and disease with green. We can move nodes, and by double-clicking, we can expand further the graph. We can expand this mutation and then display all articles in which this mutation exists. And if we expand another R mutation, we can see that they coexist in an article. Neo4j enables you to send multiple queries to the graph via CSV file. Here is an example with a query of 174,000 dbSNP mutations that takes only 2 seconds. We can see that 17 mutations have been found, and for each one of them we can see further information such as which articles contain these 17 mutations. It was a query with dbSNP mutation, but there are several ways to describe a mutation. dbSNP mutations, although they exist in literature, they might be of limited interest. This is due to the fact that dbSNP mutations are known and documented, and a new variant which is discovered might not be part of the dbSNP. Even in mutations that exist in dbSNP, some others prefer to use HGVS representation. This is because HGVS representation is more informative. The format of a complete variant description is a reference and description, where reference is a transcript of a gene and description is the position and the type of the mutation, duplication, insertion, or deletion. So because in the graph there are a lot of variants with only the description, only the second part of the HGVS, we have employed a quality control step in order to ensure the validity of the reported HGVS variant and to minimize the HGVS noise. So for each article we extract all genes that it contains. For each gene we have a list of all its transcripts, and for each HGVS mutation that does not include a reference information, we try all possible transcripts in a Cartesian product and check if they form a valid HGVS variant. The validity of these artificially formed variants is checked with variant validator. Concerning the future work, we want to apply graph analysis algorithms on the resulted graph, algorithms such as clustering or shortest path. We could use variants from CleanVar to test if Neo4j returns the relevant PubMed IDs. We also want to apply LDA on the text of the article nodes to reveal interesting topics. And finally, we intend to build a web service that accepts a VCF file and creates an explorable graph, and to create a Docker image with all components, BEP, Neo4j, variant validator. Thank you for your attention.